This class is about epidemiological uses of immunodiagnostic techniques. I'm Dr. Lau from Department of Parasitology. The learning objective includes describe the principle, techniques, standards, and recording of results, and interpretation of different immunomethods used in diagnosis of parasitic disease. Next, we look at serological diagnosis. When you talk about serological diagnosis, it's actually detecting the antigens or the antibodies produced by the human body towards the parasite. The antigen is the antigen on the surface of the parasite. It is not the parasite itself. So, in this kind of diagnosis, we do not see the parasite directly. So, we call this as an indirect detection method. To detect the antigen, we need blood samples, sometimes stool samples. Intestinal parasite can be detected using the stool sample, for example, Fusosporidium species and Entamoeba histolytica. To detect the antibodies, we need samples such as serum samples, serum or plasma, and also blood can be used. Cerebrospinal fluid can be used as well. So, in order to detect the antibodies, we can use either total antibody detection or specific antibody detection which target IgM, IgE, or IgG only. If you could remember, this is the same slides given during your year 2 class where I mentioned about different formats of immunodiagnostic tests. The first one is the enzyme immunoassay or ELISA, EIA. In the old time when it is radioactive was used, then it is called as radioimmunoassay. So this is how ELISA looks like. It consists of the plates with 96 wells, and you can read it by putting into the ELISA plates breeder. And then I also mentioned about fluorescent antibody techniques, in short FAT, where the slides with parasite can be labeled with fluorescent dye, and then it can be viewed under microscope as a fluorescent material. And then there is in agglutination assay. If you could remember, agglutination assay consists of many types as well: heme agglutination, direct agglutination, indirect agglutination. The basic principle of it is antigen and antibody. When it's mixed together, it will form agglutination, and you can see spots or dots on the membranes or the filter paper after the assay. So those with spots are those that are positive, and the one without spots are negative. And then we also mentioned about immunoprotein or Western blotting, where the protein can be run on an STS-based gel and then transfer into the membrane, could be PVDF or nitrocellulose membrane, and then the membrane will be incubated with serum which contains the antibodies, and followed by many washing steps and secondary antibodies and then detections. The last formats that I mentioned previously is immunochromatographic test, which is ICT. If you could remember, we use it for malaria diagnosis. This picture here showing that there are two bands. One is specific for falciparum malaria, another one is for other malaria species. There are many advantages of serological diagnosis tests. We usually use serological tests when we cannot detect the parasite directly under the microscope, which means the parasite is no longer in the blood, where we can use the blood samples to observe under microscope. For example, in toxocarasis or toxoplasmosis, these two parasitic infections, the parasite would not be in the body for long, in the blood for long. So once they enter the blood system, they will wiggle around to the tissue and they hide in the tissue. So once they're hiding in the tissue, the parasite will no longer be seen. But what we can detect is the antibody responses from the human body towards the parasite. This antibody will prolong in the body, which enable us to detect using serological methods. So serological method can also differentiate past infection compared to recent infection by looking at the IgG and IgM level. Another advantage is we use serological methods for congenital infection, where the infection passes on from the mother to the fetus or the baby. So in this case, we just need to diagnose the infection using the blood sample from the mother to look for the infections. And for serological tests such as ELISA, which can detect infection using a large-scale sample size, as I mentioned just now, one plate can detect 96 samples at one time. So it is very easy if you want to use ELISA plates, ELISA screening method to do a large-scale blood screenings for parasite. So if you have a lot of sample for screening, it's actually very feasible to use ELISA methods, where you can screen 96 samples at one time. So because of this also, many epidemiological tests which require a large sample size and prevalence tests of infection, usually we use serological tests to conduct the survey. So before you start using a particular serological test, there are a few considerations that you should make before you choose a test. Okay, first one is which test to choose, the selection of tests. Serological test is a variable aid in diagnosis of many parasitic infections, including some of which are very difficult to detect. There are many assay or types of tests that can be used in the market. The assay available can be ranged from standard lab techniques that have been used for decades to novel assay that are new invention as well, which develops over time. Okay, the sensitivity of this test, either in-house test or commercial test, could be very high. Some is as high as 100%. But most of the test manufacturer, the specificity and sensitivity were determined by whoever testing the kits. Okay, the value of the kits, the sensitivity and specificity may be different, and how they measure it, how they evaluate or validate the test, it could be different as well. So all these factors could interfere with the real actual value of the test itself. So to select a good test is very important before you start any diagnostic labs or start any research using serological tests. So you have to really read up about the test, either look at the literature reviews or comments from other users on the test kit itself. Okay. Serological tests for malaria, echinococcitis, Chagas disease, amoebiasis, schistosomiasis, and many parasitic infections are quite sensitive and specific, which could be used for general epidemiological purposes. So the key value to determine how good is the test is always the specificity and sensitivity. Okay. There are reports of non-specific cross-reactivity and lack of sensitivity for certain tests. Certain serological tests, for example, for nematode infections such as toxocariasis and fungoides, has been shown to cross-react with each other among the parasites. So improvement of parasitic antigen is required to enhance the specificity of these tests. Okay. When a test is developed in the lab, the in-house test, or even the commercial test, they should have cutoff point. Okay, so we can actually decide where is the cutoff point for in-house test. But commercial kits, they already determine the cutoff point, so you just need to follow whatever it mentioned in the kits. So for in-house test kit, by adjusting the cutoff point, it will affect the sensitivity and specificity. For example, okay, by putting the cutoff point for a positive result at a very low level, in this figure here, you just move it to lower level. You may capture all positive samples. Okay, the positive sample which is in the blue curve, you may capture all of them, but you may also capture some of the result which is supposed to be negative and it became false positive. In this case, the test is very sensitive, but it's lack of specificity because it capture negative result as false positive. Okay, so finding a balance is really important in this case. For an effective and usable test, we have to find a balance where is the best cutoff point. Another consideration is the persistence of the antibodies. IgG and IgM could persist in the body for months or sometimes years. So this causes some confusion in diagnosis. When the infection is not there anymore, the ELISA test is still giving a positive results. So we have to always remember that the antibody may persist depend on the intensity, duration, and frequency of the infections. Probability of detecting antibody in an endemic area is also high because of cumulative effect of antigenic stimulation on the production of antibodies. So in the endemic areas, the presence of antibodies in the body, especially at the borderline, may not be considered positive. Okay, therefore, antibody determination should be monitored where possible during the course of the illness and to be performed at least two different sampling taken several days apart or sometimes weeks apart to look at the antibody profiling. The fourth consideration is to look at the agreement among different tests. So there is still lack of accepted standard procedure for serological tests. So the antigen and the procedures used in each of the methods, they are all different. So we need to standardize the antigen and procedure used in a particular example, a project. Okay, it has to be standardized, otherwise the result will be varies as well. Okay, antibody stimulated by the parasite can be at different immunological specificity. For example, for fluorescent antibody and dye test, antibody may interact with the surface antigen of the parasites. But for other tests, the antibodies may interact with the soluble antigens. They may not have the same properties compared to the surface antigen. That's why they may give different results. So we need to really consider the differences among the tests and to pick the correct one for any particular project or diagnosis used. Those are the four considerations that you should make before you select a serological test to be used either in a diagnostic setting or in research in the lab. So we have to consider those four major indication before you start using a test. All right. So after that, we can really use a test for certain applications. So these are the application of serological tests. First, it can be used, of course, as a clinical diagnosis methods. And although the persistence of parasite antibody make it very difficult to determine which kit or which antibody to be measured during the infection, there are actually certain correlation between the antibody titers and the infection as well. So it's not as bad as that. So it still could be used as a diagnosis tools. So high titer usually indicate that the possibility of having the infection, a recent infection or an active infection. Okay. And then a rising titer in several specimens, if you collect a few specimens from the same patients, uh, the trend of the antibody titers, if it is rising and is, for example, IgM is increasing over times, this may indicate a recent infections. All right, so if you can collect a few specimens, it will really helpful in determine the infection state. So the utility of serological test as a standalone assay is limited, okay, because due to the cross-reactivity and due to the prolong of the antibodies present in the bodies. That's why it is often used as a complementary test on top of other tests performed. Okay, in this case, infection can be confirmed by many other ways For example, looking at the clinical symptoms, uh, checking under microscope examination, or doing some radiology scanning, biopsy, and each other, or even PCR can be performed, okay, to complement the result of serological tests. Okay, in this table here, showing that one of the example of laboratory diagnosis of Toxoplasma gondii. Usually, there are many methods used to combine together to give confirmatory results. Okay, for example, we can use microscopy to look at the tetrazoid or the cysts in the blood, although it's very hard to find, but it is one of the possible way. And then, of course, we can perform serological tests. There are many tests available for Toxoplasma gondii, for example, ELISA, IFRT, IgM, ISACA test. All these can be used as one of the serological diagnostic tools. And then back up with PCR, which is very useful, and back up with clinical diagnosis such as MRI, CT scan, which can be used to determine whether the parasite is present in the brain. And also, there are many other methods like animal inoculation, skin tests can be performed as well to confirm the infection. Serological test is especially useful in large-scale screening of samples. When you have large amount of samples, this is a way to use for diagnosis, because just like ELISA plates, it can actually do 96 samples at one go. So it's very suitable to screen a large amount of samples, okay, compared to PCR methods, for example, which need to be performed in tubes, one tube at a time. So it may be very time consuming and you have to handle many samples at one time. But for ELISA, you can do 96 samples in just one single plate. Alright, so because of that, it's very useful for us to use serological tests to determine the prevalence of disease in populations, or to assess the prevalence of antibodies of a specific organism, and also to assess the protective titers of antibodies used in populations. So this is particularly useful in deciding which antigen is suitable for vaccines. This is one of the examples showing that how a serological test is useful to determine the antibody protections level in populations. This study was carried out in Kenya in malaria endemic areas in 87 children. All right. So the authors used ELISA to determine the titers of antibodies to three antigens: AMA1, EPA175, and MSP119. These three antigens. So they look at the antibody titers towards these three antigens and check whether the presence of antibody could protect the children from malaria infections. All right. So this is the table showing the results. So for AMA1, the risk of children getting the infections after having AMA1 antibodies is about 77%. Okay, the value here is 0.77. Means 77% of the children with the presence of AMA1 antibodies in their body, they will still get malaria infections. Okay, whereas in EPA 175 and NSP 119, the relative risk in children with high titers or antibodies towards these two antigens were quite low. It's about 48% or 46%. So these antibodies, the presence of antibodies towards EPA 175 and MSP 119 could actually protect 50% of the children for getting reinfections of malaria. So this is how useful is the ELISA test performed in order to show that whether the antibodies can be protective. Another application of serological tests is to look at surveillance of eradication programs. Okay, so serological methods have very practical applications in the surveillance of eradication program such as immunization program or vector control program. So it can be used to determine whether a transmission of a disease has been interrupted or reduced. Okay, just look at the title level. Okay, following the establishment of the baseline data in the populations, the interruption of transmission could be reflected in decrease of antibody titers. Okay, the effect of eradication program is most suitably measured by study of the antibodies in the very young children. So by looking at the young children populations, we can actually determine whether the eradication program is successful or not. Okay, if it is successful, you can see that there should be a decrease in antibody titers, especially in the young children, because the infection is no longer there, so the children were not infected and the antibody titers has thus been reduced. Serological tests can be used to determine the level of endemicity. So by plotting a seroprevalence curve, we can know whether certain areas is low or high endemic. In low endemic areas, a bimodal distribution curve can be obtained by plotting the frequency of the infections versus the antibody titer. In this curve, the first mode is in the low titer range of the curve. This range may include non-specific reaction with the sera of non-infected individuals or individuals in the population with very low antibody titers. So majority of the people in these areas is having very low level of antibody titers. The second mode may represent specific parasitic antibody levels measured with the antigen and broad. So in this case, the test can actually detect the infection at higher level, which we can consider them as active infection. So the shape of the distribution help us to establish a diagnostical meaningful titer for this population. Example in this curve showing is 1 over 128 dilution was determined to be clinically significant value. And this curve is helpful in determining the significance titer measured in the sera from this group of patients to decide whether one individual is actually positive or negative. Let us look at the curve closely. So this is the bimodal curve showing in low endemic areas. So the first curve mode here is non-specific binding or individual with very low titer. These people here may not present with symptom. They may be having very low level of infection or there's no infection at all. And then the second mode here is showing the specific parasitic antibody bindings, where the titer in this case is about 1 to 256. So this value indicates that these people in this group here is having the active infections. So by plotting this curve, we can determine that the titer 1 over 128 titers is a positive value. Anything above this value can be considered as positive. But the population having the positive value in these endemic areas is quite low. So we can decide that this is in low endemic areas. In areas where the parasite is absent or with very low prevalence, a single low titer peak or mood resembling an exponential curve can be seen. So this is where the single peak at the low titer is present in majority of the people in these populations. And then a high titer values at 1 over 256 here are too few to create a second peak or mode. So there's no peak at all at the second mode here. So in this curve, it represents an areas with very low endemicity. So in highly endemic areas, a unimodal curve is obtained. This is how the curve look like. Most of the majority of the population are having very high infection with very high titer value. Okay, in this case, most of them is having a titer value at about 1 to 500 titers. So by titrating serum collected from a population, one can determine the prevalence of a parasite antibody. The type of the frequency distribution curve can use to determine the level of endemicity. This paper here shows that the indirect heme agglutination test is used as a sero-epidemiological technique for malaria infections. By employing the serological test, this method can use to measure level of malaria endemicity, can also measure malaria transmission interruption or reductions, and can use to look at the seasonal change of malaria transmission, and so many other things can be found using the serological methods. This is another example of paper using immunodiagnostic tests for epidemiological studies of cysticercosis or daniosis in vesicle. So cysticercosis is acquired when swine ingest human feces contaminated with Denisonium eggs, and human in return can also develop cysticercosis after swallowing the Denisonium eggs. So human neurocysticercosis is considered a public health problem in Mexico and several other countries in the world. So the development of immunodiagnostic techniques has promoted the conduct of sero-epidemiological studies worldwide. So this paper uses the serological test to look into the risk of getting cysticercosis. So it was shown that the close proximity with the tapeworm carrier is the main factor of acquiring cysticercosis. So this immunotechnics has helped to pinpoint the transmission foci so that appropriate and effective intervention can be applied. Well, that is the end of the lecture today. Before we end this lecture, please take some time to answer the following questions. If you listen to the lecture carefully, you should be able to answer all these questions by now. These are a few references you can refer to. Thank you. If you have further questions, you can always contact me by this email. Bye.